Alright guys, I am making this particular video at home, so I hope this works out okay. I hope my computer is actually recording this, so we shall see. This is going to be our last lesson of the semester, and it's going to be all about biotechnology. And this is something that we could probably spend a lot more time on, but with it being the end of the semester, we're just going to kind of talk about some of the basics. And so we're going to start off with, what is it? And if you kind of break down the word, bio is life, technology is like using tools, and then ology is the study of. So this is all about using living things as tools. And really what we're trying to do is we're trying to modify essentially living organisms in a way that makes them useful to us. And so one of the main things we'll do is we'll kind of modify these living organisms to produce new compounds or to perform new functions. And really, it's not just a science discipline. I mean, it's something that involves many disciplines from engineering to computers, especially in today's modern biotechnology techniques. A lot of it is just you put the information in computers and they spit the information back out. But you can see that there's a lot of biology branches involved. It's very heavy in genetics, microbiology. Your microorganisms are used very heavily in biotechnology, things like bacteria and yeast. We use things like viruses to kind of move genes around, and so we do need to understand a lot about diseases. Obviously, molecular and cellular biology, that's going to be like the study of DNA and proteins and things of that nature. So quite a bit of different subjects involved. Now, really, it sounds like it's kind of techy, but the reality is biotechnology is something that's been around probably since the beginning of humans, because we've kind of utilized ways to grow crops better or produce better animals. This is something that started way back when we just had an agricultural type society. And so some of these practices have been around quite a while, like artificial selection. Here in a minute, we're going to talk about selective breeding, which is essentially artificial selection. And like I mentioned, we use bacteria and yeast for quite a lot of biotechnological techniques. And things like bacteria and yeast have been used to produce food and alcohol and different products like that for a very, very long time. Now, some of the benefits of biotechnology, we use them very heavily in medicine, and this is not a list of everything. We could probably do slide after slide after slide of the benefits of biotechnology, but we do use them to produce medicines like antibiotics. We produce human proteins like insulin using bacteria. There are gene therapies. There are all different types of making vaccines and things of that nature. We can use them in what we call bioremediation, and that's what this environmental benefit is. We can use bacteria and microorganisms to clean sewage. We use that in water treatment. We can use it to clean up oil spills. There are bacteria that have been genetically engineered to kind of consume and digest that oil. It's very heavily used in agriculture, and we're just continuing to advance in the things that we can do there, but this has been used for a very long time. And then we use it to make foods. We use it in industry and manufacturing. And there's just a lot of different ways that we could utilize these living organisms. Now, what we're going to talk about for the rest of the PowerPoint are these various biotechnology techniques. And like we've already mentioned, some of these are not new. Some of these have been around since maybe like the 1950s or a little before. A lot of the techniques were discovered in the 1970s, and then they've just continued to progress. So I do want to remind you that Watson and Crick did not build the model of DNA until the 1950s. And so a lot of our advances have come after that, but we've been utilizing this manipulation of living organisms for quite some time. And I'll be honest with you, the lesson that I'm teaching you today is really outdated. We're not really talking about a lot of the most current advances that you have in biotechnology. I think it'd be pretty awesome if some of you guys would go into a field like this. It is a field that is constantly growing. As we learn more about DNA and genetics, the more we can do in biotechnology. And so there's a lot more out there that we're not discussing. And so I just want you to be aware that these are just some of the basics. This is something I wanted to include. I know it's very, very small, but this is just a kind of an overview of a timeline of biotechnology. And you can see it goes all the way back to BC times. And even though this particular one stops at 2013, we are continuing to see advancements all the way up to 2025. So this is not a field that is slowing down. It's a field that's going to continue to grow and change over time. All right, so we are going to start with something that's very, very old, and that's selective breeding. This is something that human beings have done for a very, very long time. And sorry, I've lost my mouse. Not sure where it went to. But selective breeding is where we choose the traits that we want. And we see this very commonly in agriculture. If you want the cows that produce the most milk, or you want the pigs that have the best meat, or you have the plants that are the most disease resistant. And so when you cross plants with traits that you want, that is an example of artificial selection or selective breeding. A lot of times this involves a process known as hybridization, which is crossing different organisms. So we saw this back in genetics. A hybrid is a cross between two purebreds. And so this way we can get the best traits. So like disease resistant with high yield, or maybe disease resistant with good meat or something like that. So we've been doing this for quite some time. There's nothing new about that. We also just do it kind of for fun in terms of like dog breeding. All of your dogs are the same species, but yet they look so different. So we kind of take traits that we want. Like I have a dog that doesn't shed, and he's small. He's a miniature. And, we can take horses for racing, or we can take different animals and cross them like the liger there. And we can make plants that are the best of both worlds by crossing two that have traits that we desire. All right, something else that we can do is we can actually trigger mutations, and we know that mutations have kind of a bad rap. But remember, not all mutations are bad. Sometimes mutations can lead to good things. Now, we don't go around doing mutations on humans because that would not be ethical. But a lot of times with bacteria or microorganisms or plants, they will intentionally induce a mutation in hopes of getting a beneficial trait. It's pretty common to make plants polyploidy. One of the things you're going to do for this unit is you're going to extract DNA from a strawberry, and strawberries are polyploid. A lot of the fruits and vegetables that you see in your grocery stores are polyploid. For some reason in plants it makes them bigger and better, but in animals it's fatal. So we don't really do this a lot with animals. All right, another thing that's pretty common with biotechnology is genetic engineering. And this is something that's been going on for a while. After the discovery of the DNA structure, I know it definitely started by the 1970s, probably before that. But this is something that's been around for a long time, and it's anytime you intentionally make changes to DNA. Now, the first thing you have to do is extract it. And this is one of the things that you're going to do for a lab is you're going to extract that DNA from a strawberry. And then the next thing we can do to genetically engineer is we can cut the DNA. Now, we obviously can't visualize it. We can't see it. And so what we use are these special enzymes called restriction enzymes. And what they do is they always cut DNA at a very specific sequence. And when they cut, they don't cut like a typical straight across the DNA. When these restriction enzymes cut, they cut in a staggered pattern. And so what happens is you see this little tag called a sticky end hanging off. And so the basis of genetic engineering is you take two DNA sources. You could have one from a bacteria and one from a human, or one from a plant. If you cut them with the same restriction enzyme, they will leave the same sticky ends. And so what we'll do with that is we can get DNAs to join up. And we'll talk about that here in a minute. Another common process that's done in a lab is called gel electrophoresis. And this is a separation technique. So once again, we can't see these DNA pieces. And so you can take a piece of DNA and you can cut it up into smaller pieces with those restriction enzymes. And then we can use gel electrophoresis to separate them out. And so what you do is you literally kind of inject them with the pipette into a gel. There's these little wells. And if the DNA is cut differently, they will have different size fragments. Now, it's based on two important concepts. Number one, DNA has a negative charge. And so when we apply a voltage, which is where that electrophoresis comes in, just like we have a positive and negative end on a battery, when we apply this voltage across the gel, you place the DNA on the negative end, and because it's negative, it will move toward the positive. And so it causes these little DNA pieces to exit the well and move toward the positive side of the gel. Now, the other important concept behind gel electrophoresis is that the smaller the piece is, the faster it is. And so I've always used the example of if you had to go to the office by yourself to pick something up, you could go and get back very quickly. But if you had to like hold hands with everyone in your row and go to the office and pick up something and come back, you would be slower. Or if the whole class had to hold hands and then go to the office, you'd be even slower. And that's the way these fragments are. The bigger they are, the more resistance they have moving through the gel. And so over time, the DNA pieces will separate out by size, and that's how the separation occurs. So you apply the voltage, they move toward the positive end, and then these little tiny pieces of DNA that have been cut up will separate out based on their size. The smaller ones move farther, and the larger ones are closer to the wells. And then we can use this process, or we have used this process, to do DNA fingerprints. Now, this is one of those areas where it is advanced considerably. They have much faster ways and other ways to kind of sequence DNA very quickly. But kind of our first DNA fingerprints came from this basic concept of gel electrophoresis. And so what you can do is you can create a fingerprint, not like on your fingers, but with DNA. And essentially what it does is when we separate those DNA pieces, it creates these unique DNA bands. So what they're going to do is they're not going to use DNA that's the same in everybody. We're all human. Our DNA is more alike than it is different. And so what they'll do is they'll take these sections that are varied. They take a lot of these repeat sections and these non-coding sections, and they'll cut them up. And then if you're related to somebody, you'll have similarities. If you're not related to them, they will be more different. And so you cut the DNA, you run it by the process of gel electrophoresis, and what you get as the end result is this unique banding pattern. And so you can see, like, here's the mom, and here's the child. The child is going to be a combination of mom and dad. And so like on this one, if you're trying to figure out who's the dad, it's going to be someone who can give the child these similar markers along with that mom. Over here is another example. So you have a crime scene, you have 3 suspects, and you can find the one that has the same banding pattern. So that's what a DNA fingerprint is all about. Another very important method is something called PCR. And back when biotechnology first started, in order to do any kind of DNA testing, you had to have a very large sample. Like you would have to have at least a quarter size or more of DNA, of like blood or body fluid or something like that. Well, polymerase chain reaction came about in the 1980s, I believe. And what it is it's an amplification technique. It's a way to make many copies of DNA very, very quickly. So let's say you had a little tiny speck of blood left at a crime scene. What they can do is they can take the little bit of DNA that's in that sample, they can run it through PCR, and they can make a lot more copies. And so what they do is they take really kind of 3 main components. They take the DNA sample that they want to amplify. They use DNA primers. Remember that DNA polymerase needs something to build off of. They use the enzyme DNA polymerase, which is our builder, and they put a whole bunch of spare nucleotides. And so kind of what it involves is first you have to open up the DNA, you have to change the temperature, you kind of warm it up and then you cool it back down. And then what they do is they mix in these primers, these nucleotides, and DNA polymerase, and then it will just go to town, making copy after copy after copy. After a few cycles, maybe like 30 cycles or so, you can have millions of copies of this little DNA fragment. And so this was really important, like for investigations where you didn't have a big sample to begin with. All right, another technique, and this is one I kind of touched on earlier, is recombinant DNA. And so this goes back to cutting that DNA with restriction enzymes. And so all recombinant DNA is when you take DNA from two different sources and you recombine them. So what we're going to do is we're going to cut two samples. If we use the same restriction enzyme, it will leave those sticky ends. Okay, so as long as you cut both samples with the same enzyme, remember it only cuts at a very specific location, it'll leave these little sticky ends dangling. So like in this case, you could cut a gene out of human DNA, and you could cut a bacteria chromosome or plasmid with the exact same restriction enzyme. And if the sticky ends are complementary, they will join up, because we know that this DNA is based on the base pairing. So if you have these sticky ends that match, they'll join up. We use the enzyme ligase to kind of glue them together. And what you end up with is a DNA molecule that has information from two different sources. Now, the next thing we can do with that is we can undergo a process of transformation. And so this is where we see bacteria used quite a bit, because people don't really get upset if bacteria die. They're really easy to work with. They grow very rapidly. They're kind of easy to manipulate their DNA, and nobody's upset if we harm the DNA or DNA die in the process. And so what we do is we take these little circular pieces of DNA called plasmids. We mentioned these a little bit back when we covered prokaryotes. And so you could take a gene from any organism and you could get it inserted into the plasmid. And then if you get that plasmid into the bacteria or into another organism, that's what transformation is all about. It's about introducing DNA from one organism into another. So let's say that this little red here was a human gene. You get it into the plasmid using that recombinant DNA technology. You then get the bacteria to take in the plasmid, because they'll naturally take it in from their surroundings, and we can kind of manipulate them to get them to be more likely to take it in. And once this little plasmid is in this bacteria, the bacteria is going to copy it, just like it would its own genetic material. Also, that bacteria is going to replicate. And so we can make copies of that gene. That bacteria can express that gene and make the protein. And this is one of the ways that we create bacteria that literally produce human insulin, or they literally produce human growth factor or human clotting factor. We have bacteria in labs that are just pumping out human proteins because we've transformed these bacteria with a human gene. And so it does involve those plasmids. Once again, we have to insert the gene into the plasmid. That's the recombinant molecule. And then once you get bacteria to take these in, we use vectors. The plasmid is a vector. We sometimes use viruses as a vector. But once that gene gets into that living cell, we now have had transformation occur. And this is how we create transgenic organisms, which are organisms that contain genes from other species. So if you put a human gene into a bacteria, that's a transgenic bacteria. Now you can make mice that glow by putting, bioluminescent genes into a mammal. We could put genes from one plant into another plant. We could put genes from, one animal into another animal. A lot of times we're transforming bacteria, but we can do other animals. It's not really done a lot with humans. That was once again, when you start getting into humans, we get into some issues with ethics. But we can use this to make human, medications. We can use it to make foods that are better or healthier. We can make better crops. And this is what we essentially call GMOs, y'all. You hear this in the news. Pretty much a GMO is a genetically modified organism. And so that's essentially what a transgenic organism is. So here's some examples. You could put a gene into, pig embryos that make them produce leaner pork. You could put genes for milk production into different animals and help with their milk production. You could pick a trait that you wanted in a plant, like we have disease-resistant tomatoes and things like that, and we can make better crops. Now, when it comes to the humans, there is something called gene therapy where you could get a healthy gene into a person that had a faulty gene. This does happen, but it's one of those things that anytime you're involving humans, you have a lot more medical trials that you have to go through. So there are some current gene therapies, but a lot of them are still in the development stages. They're not yet fully developed. So we do have some out there. A lot of times, what they'll do for humans is they will insert the healthy gene into a virus, not one that's going to be harmful, but that virus can then get that new healthy gene into the person, and then hopefully that person can make the protein that they were lacking due to their disease. So there's lots of potential with gene therapy with curing certain genetic disorders that could not be cured before. So here's kind of what they do is they take the patient's cells. They're going to use different vectors. There's some that aren't viruses. There's some that are viruses. They get the new genes inserted, and then they put that person's cells back into them. Hopefully those cells are going to multiply, and then they'll produce the protein from that healthy gene, and then that can, take care of a deficiency that a diseased person might have. All right, one thing that we do a lot of with DNA technology is analyzing human DNA, and this is something that has progressed greatly. There have been a lot of different sequencing techniques that have been established. Back in, I want to say it was maybe in the 90s, they started doing the Human Genome Project, and they were literally trying to map out every letter of the human genome. Now, this is something that I need you to correct in your notes, okay? Notice I bolded it here. I realized I did the note sheet after I was, or before I was finished editing the PowerPoint this time. And so there are roughly 6 billion with a B, okay? 6 billion base pairs in the human genome. And so this is looking at our 46 chromosomes. If you were just looking at one set, it would be 3 billion. So we're talking a lot of A's, T's, C's, and G's that make up our DNA. And so what we're trying to do, and they're still currently working on, is we have the human genome mapped out. We kind of have this basic set of billions of letters, and now what we're trying to do is decode it. Which sections are genes, which sections are regulatory sequences, and this is something that we're continuing to do and learn more about and identify disease genes and where DNA can go wrong and what it causes. And so there's still a lot of studies going on because it's kind of hard to decipher 6 billion letters quickly. And so a lot of times we can use this to identify certain diseases or identify the genes that are responsible for different proteins. And the more we can learn about that, the better off we'll be in a lot of different areas. So here's just a very brief example is if they want to find a certain DNA sequence, what they can do is if they know what they're looking for, they can create a complement called a probe, and they tag it with radioactive isotopes. And then when they mix that probe with the DNA, it'll attach to the area of interest. And then this radioactive isotope will help us visualize it. So they can kind of find certain sequences of DNA that they want to do. A lot of this is computer generated. And so I don't even know how to read all of this, but this is showing you 4 different sequences of DNA, and it's showing you those bases. And so you can compare organisms, you can compare humans, you can do this for evolutionary study. There's so many different ways that we can utilize these DNA sequences and learn about life. Another technique is cloning, and a lot of times when people think about cloning, they think about cloning whole organisms, but a lot of times what we're cloning is just cloning genes. Anytime you're making a genetic copy, we're making a clone. And so it's very common for us to do what's called molecular cloning, and that's where we just clone individual genes. And so what we do is we did like what we saw earlier. You take a gene and you get it into a bacteria. Those bacteria reproduce like crazy. And so we can go from one copy of that gene to millions of copies of that gene in a very short period of time. And then we can use that to study those genes and just have as many samples as we want of them. Now, it is a lot more complicated to actually clone a whole organism. This is where you take an egg from a female and you take out the nucleus. And so essentially, you've removed that haploid set of genetic info. And then you take a full diploid nucleus from the organism that you're wanting to clone and you put it in the egg cell. Now, it's really important to understand that you can't clone an, an animal or an organism in the current state. So, like if you wanted to clone me, Miss Tier, it wouldn't come out as a 44-year-old Miss Tier. It would have to go through all of the life stages. It would have to start as a single cell with a diploid set. Like they could take one of my skin cells and remove a nucleus and then put it in an empty egg. But then that egg would have to go through cell division. It would have to be implanted into a surrogate. It would still have to go through all the stages of development and be born. It would still be a baby and have to go through all of the different life stages. And the reality is, even if you clone it, they may have genetically identical DNA. But remember that part of what makes us us are things that we're exposed to, our environment. And so when you create a clone, it's not going to be the same, like in terms of personality. So like you can clone your pets. If you had a pet that passed away, there's companies that will clone your pet. But if you did that, it wouldn't be the exact same dog or the exact same cat, because part of what made that animal what it was environment. And so the first that we know of, the first animal that was cloned was Dolly. It was a sheep back in 1997. And one of the things they found is that a lot of times these cloned individuals have a lot of genetic defects, which, keep in mind, that DNA is as old as the sheep that they cloned. So if they took it from a sheep that was several years old, then that DNA is already several years old, and it's going to just continue to be copied. So it's not like really starting from scratch. So it is possible to make these genetic copies, but we do the molecular cloning a lot more. So here's the molecular cloning. You get a gene into a bacteria, and then the bacteria does all the work. And then here's an example of how you could do the cloning of a whole animal. All right, and the last thing we're going to talk about today, and like I said, we did not cover all of the techniques. We actually covered quite a few. Many of the ones we covered have been around for a while. This is definitely a really cool field, and I wish we had more time to spend on it. But one thing that always comes into play is it ethical? It's really important that as a scientist, you have to take into consideration of just because we can doesn't mean that we should. And so anytime we get into biotechnologies, a lot of times ethical issues are raised. A lot of times people have issues with GMOs. They definitely have issues like it's not legal to clone a human. Anytime we involve humans and human life, it brings in this question of, is it morally right? And obviously that's different for different people because we have different morals and different viewpoints. And so one of the things that we do have to address, though, is, as we make these scientific advancements, we have to kind of consider, is it okay? Is it the right thing to do? Is it something that is morally upstanding? And so that is definitely an issue that you will face, is with all of these biotechnology techniques, is it okay? Is it something that we should be doing? And so always keep that in mind, that just because we can doesn't mean that we should. But we've actually had a lot of advancements with a lot of good benefits, and it doesn't mean we shouldn't continue to learn. And that is going to wrap up our lesson, our very last lesson for our semester. You're going to do a couple of activities. And in terms of testing, you guys are not going to have a test over the unit, but you will see questions about this material on your final exam. And hopefully I'll be around soon to see you guys. Hope you've had a great semester, and enjoy the activities that you're going to be doing over the next couple of days. Talk to you soon.